I must say that having mentioned sports and the Nobel Prize, I feel compelled to begin with a light anecdote and say that when I was asked a couple of times what I was going to do with the money, I remembered that a similar question was posed to the great Irish footballer, which some of you may know, either those who are my age will know from having watched him, and younger people from his reputation, Georgie Best, played for Manchester United for a dozen years or so. And he was the first sports icon who made money not only from his salary, but from endorsing products, really began a whole era. And he died penniless many years later, with cirrhosis of the liver. And he was asked on his deathbed, more or less, how, having earned so much money, did he end up penniless? And so he said, well, I'm going to explain it to you. He said, I spend a fortune on fast cars, women, and booze, and the rest I squandered. And so that's what I wanted to do with the money, but my wife had other plans. So let me try and walk you through a story about a more serious story about circadian rhythms, and this is the point of departure, the definition, if you like, that these are self-sustained rhythms. That means they don't need a light-dark cycle. In constant darkness, under constant temperature and humidity conditions, this metronome-like clock keeps on ticking. And it does not have 24-hour periodicity under those conditions. It may be 23, it may be 25, it varies with the species, hence the term circadian or circa dia from Latin, about a day. And the way it gets to be exactly a day, the way we get to be on track, where every day is the same as the day before, is through communication with the light-dark cycle. Light resets the clock every day back to zero, a clock that runs in us a little slow, as I'll tell you a bit more about shortly, and in other animals runs a bit fast. And the purpose of the clock is really anticipation. Knowing what is going to happen. So this is simply described by the early bird gets the worm, and the early worm avoids being eaten by the bird. If you know what's going to happen, you're much better off than if you simply recognize it as it's happening. It probably also functions to govern what's commonly called internal coherence, namely a whole bunch of stuff happens internally, biochemical reactions, and it's more efficient, it's better if A happens before B, C, D, rather than them all happening at the same time. And so this whole system arose long ago, as the slide title indicates, and the rotation of the Earth, this continuous light-dark cycle, 24-hour light-dark cycle, is the oldest environmental cue, or as it's called even in English, Zeitgeber, but in English, Zeitgeber, way before the Earth had its current gaseous constitution, before nutrition for animals was anything like what it is today. This kept on turning, and life was exposed to it. So the oldest known clock is in cyanobacteria, photosynthetic organism, and cyanobacteria are responsible for oxygenating the atmosphere starting more than 2 billion years ago. And there's a very, very well-described clock in cyanobacteria. And importantly, that clock is completely different from the clock that is present in plants and the clock which is present in animals. So these are three completely different systems. They operate on different principles. They're different timekeepers. And the important point is that this has arisen multiple times in evolution. It's so important that different organisms have reinvented it over and over again because of the value of anticipation to survival of the organism. So I'm going to begin and pay homage to one of the giants of our field who died earlier this year, Sidney Brenner. And just to put things a bit in perspective, this is a quote from him. And I will use this as a touchstone at a couple of points in the lecture. And in particular, I want to emphasize that discoveries in this sense include things that are by accident, for example, as you indicated, Fleming's discovery of penicillin. So you can stumble upon something, it can be very important, you might be think your way through it, but on the whole, I of the belief, religious almost, that ideas are cheap and most of them are wrong in science. We continually think you understand something or you have a good idea, and it's almost always wrong. And so this is what we call in the States hypothesis-driven research, and when it's strictly idea-based and not experiment-based, I'm not a great fan. And so this is a segue, really, to tell you about locomotor activity. This is a very old slide, as you can gather, because there's a chart recorder in it as opposed to a personal computer, and that chart is keeping track of the revolutions of the cage, of the running wheel that the hamster or the mouse is in. And what you notice is that in constant darkness, and there are 7 days, 7-day record on the top, that every day the activity events of the rodent starts and ends a little earlier than the day before. In other words, that rhythm is less than 24 hours in darkness. And remarkably, and this is the remarkable aspect to it, that every animal is bang on exactly the same with the tiniest little variance. And we'll discuss this in the context of humans in a little bit. So in other words, almost unprecedented in the area of animal behavior, you don't have to do a population study and average a population. Each individual can be identified and is almost exact replica of every other individual. And the second piece of this slide indicates what happens in a light-dark cycle. That is, the animals are exactly 24 hours. So light is resetting that slightly fast clock every day. And you'll notice also this animal is nocturnal. That is, the activity ticks coincide with the dark phase. This is something you might ask about in the question period. Mice and rats are pharma, biotech's major go-to animal for drug discovery. And there's some question about whether using a nocturnal animal for this when you're interested in a diurnal species like us is whether that's necessarily the best strategy. So that's just something to keep in mind. So this characteristic, the behavioral rhythm, the free-running period, is the phenotype, and the mantra in genetics is like in real estate, except, of course, instead of location, 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 it's phenotype, 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 right? Okay, so this is what you do with a rodent, and now what do you do with a fruit fly? And this is how fruit fly circadian rhythms are assayed, and this doesn't give you the full picture, but the general idea is we have hundreds of tubes like that with a little bit of food in the bottom and a stopper in the top, and there's one fly in each tube, and around each tube is an infrared light source and a photocell on the other side of the middle of the tube. And that's a beam break device used commonly in animal behavior. The fly runs up and down the tube, and it makes lots of events when it's running, and when it's resting or sleeping, there are no events, and you get very good rhythmicity out of such a fruit fly locomotor activity assay. And that was the assay in 1971 that Ron Konopka, a graduate student used in a genetic screen with his advisor, Seymour Benzer, to look for mutants of fruit flies that would show altered circadian rhythms. And what's shown on the three traces are a wild type pattern, ticks of activity, rest, ticks of activity, wild type separated by about 24 hours. And then they isolated a long period strain. What's on the top here? Oh, sorry. They isolated an arrhythmic strain, which are insomniacs. They just have no pattern, discernible pattern. And then a short period strain, which is 19 hours, and a long period strain, which is 29 hours. This is a landmark paper in behavioral genetics, arguably the most important paper in the field. It's a decade or so before I even knew what circadian rhythms were. And this paper received 10 citations in the first decade after it was published. So for those scientists among you who chase rapid notoriety in Nature, Cell, and Science, be aware. There's other ways to proceed. So for the general audience, let me say a word about the purpose of genetics here. Why do you get mutants? Why do you do this? And the answer, as written in the first couple of sentences there, is that this is the only way you can actually dive in and perhaps access what is otherwise a mysterious, almost a mystical process. These flies or these rodents having this rhythm. I mean, what do you do? Where do you look? What can you possibly do? And so getting mutants in the proteins, in the genes, and then to access the proteins which affect this process gets you potentially to the mechanism, to the gears of the quartz crystal, depending on whether the relative analogy is analog or digital. And this concept of genetics is very different from you versus me. You know, is my rhythm different from yours or something like that, or nature nurture. And I try to make the point for general audiences that even nurture is heavily dependent on genetics, because everything needs proteins, and proteins are made of genes, and the genes can vary from one person to the next, and so there's no escaping the force of genetics. And lastly, the last point on this slide is doing a genetic screen looking for mutants which affect a certain process is really the opposite of hypothesis-driven research. It is a fishing expedition. But the great thing about fishing is you never know what you're going to catch, and you know, there's always the hope for something fantastic. So all of this work led in about 1998, a whole collection of circadian rhythm genes were identified in mammals as well as flies, starting with that period gene that Konopka and Benzer identified genetically, and as Torben alluded to, that my lab and Jeff's at Brandeis and Mike Young's lab at Rockefeller initially cloned. And there were a half a dozen genes in the early days that were cloned, and they led to this view of the core timekeeper. And what it is, and I think this is still true to this day, is a transcriptional feedback loop where those yellow proteins, clock and cycle, actively promote the transcription of the period and timeless messenger RNAs. And those messenger RNAs and the resulting proteins, the period and timeless proteins, feedback and inhibit their own synthesis. They turn off their own synthesis. And that creates an oscillation with the appropriate delays that oscillations require in order for things not to come to equilibrium. So this is a molecular biology feedback loop. You make some stuff, you wait a while, the stuff turns itself off, the stuff decays, and when the levels get low enough, 24 hours have gone by, and the whole process starts over again. And as the cartoon indicates, this process is not only identical between flies and mammals, but these are orthologous proteins. These are the exact same proteins that existed, that function in flies and mammals. And important to this story is the fact that some of these key genes, shown here as the period messenger RNA itself, were discovered initially by Paul Harden in my lab to undergo oscillations in level. So, in other words, it was the model was heavily influenced by the fact that this RNA itself undergoes oscillations in the head of the fruit fly, making a dynamic process of synthesis and degradation very, very compelling. And just to show you something a little more modern for the scientists in the audience, and I won't belabor the actual experiment, but this is a chromatin immune precipitation or something very similar to it. It preceded this a little bit, but this is a time course of the clock transcription factor at 6 times a day from the morning to the evening here. And what one can see on the enhancer, this EBOX element upstream of the timeless gene, this positive transcription factor goes on and then comes off as a function of time of day, driving the transcription. And the repressor protein, the period protein, also joins the chromatin, but does so in a delayed fashion with respect to the activator. And in fact, the repressor participates in ripping the activator off the DNA, and that's in fact part of the mechanism by which this gene is transcribed, and then that transcription wanes. And just for completion's sake, I need to say that this timekeeping, this basic principle, again, both in mammals and in flies, involves post-transcriptional regulation as well. In other words, this is not all gene expression at the transcriptional level, but there are kinases, there are phosphatases, modification enzymes, a number of other factors which are involved in and contribute in a major way to the timing process. So that's sort of the general picture, and I want to segue at this point by simply reminding you or summarizing by addressing why is this system identical between fruit flies and mammals. And I was telling this to a journalist today that this implies that our common relative, that is fruit flies and humans, are cousins, distant cousins, but long ago, about 550 million years ago, we had a common ancestor. And the only logical explanation for the fact that we have the same system running as fruit flies is that our common ancestor had it running 550 million years ago, and then we have both inherited the same system. And of course, there's little tweaks subsequently, but basically, it's the same process. I was on a humor radio show in the U.S. Wait, don't tell me, for those of you who have spent time in the U.S. on PBS, and I was explaining this, and Paula Poundstone, one of the comedians in this show, when I said we were cousins, she said, "You mean I've been killing my cousins with a newspaper all these years?" All right, so the segue here is to try and address for you what are the challenges moving forward. There was a past, present, and future in my title, and I want to try to address for you what's interesting about all this. Why should anyone pay attention? Isn't this all wrapped up? And I need to emphasize the fact that we actually don't know what accounts for the timing. That is, what in biochemical terms, what's the rate limiting step? Why in humans 24.25 hours? Why not 23 in humans, or 26 or 27? Somebody would say, what's rate limiting? And the answer is we don't know. And that's for the lack of in vitro reconstituted systems, structural information, etc. And related to that is a very mysterious and interesting phenomenon that I don't have time to dwell on in any detail, but it's temperature compensation, or what a modern person would call temperature independence, I think. The period of circadian rhythms changes not at all with temperature. So a fruit fly at 18 degrees and 29 degrees has exactly the same circadian period. In other words, the Q10 is 1.0, or nearly 1.0. And that's extremely unusual in biology because enzymes go more rapidly at higher temperature than they do at lower temperature, and everything in biology goes faster at higher temperature, including cyclical processes like the cell cycle. But the circadian cycle doesn't change at all, and this is even true for mammals. And you would say to me, oh, that's silly because mammals control their body temperature. But you can take a tissue out of mammals, take it away from the hypothalamus and the temperature regulatory system, put it in culture, measure the circadian period in tissue culture, no change with temperature. So there's something intrinsically temperature insensitive. And by the way, this makes great heuristic sense, because if you're a fruit fly outside and a cold front comes through and it's 10 degrees colder, you don't want your timekeeper to be messed up. You still want to know what time it is. But how does it do it, right? So we don't know. So, as I said, it requires biochemistry and structure. So the second challenge, now more pragmatic one, is health, right? How can we leverage this information to improve the human condition? And listed here is a bunch of stuff, which some of which will be make great sense to you, like jet lag, which makes great sense to me right now, having arrived this morning, and others maybe you know less about, and I'll touch on a couple of these. And the point of departure for this section is the fact that the clock, the circadian clock, has its fingers in just about everything, in all aspects of physiology. In other words, that there's as this slide indicates, from sex on your left to cancer on your far right, everything is impacted by the clock mechanism, and the question is why, right? And so I'll before getting into one or two of these in more detail, I'm going to try to give you a simple explanation for why so much of physiology is affected by the clock. And so the sort of simple answer is this little transcription feedback loop I described for you. It is operating in every tissue in your body, virtually every tissue. Kidney, liver, skin, heart, muscle, brain, everywhere. So this thing is ticking away everywhere, and in every one of these tissues or cell types, there are hundreds or thousands of messenger RNAs which are under its control, which oscillate, for example, directly or indirectly affected by the clock. We know that that is dependent on the clock mechanism, because if you assay any of those tissues in a mutant which doesn't have the clock, these core components ticking, then all those messenger RNA oscillations disappear, right? So the clock is essential for that, and as a consequence of this, when you add up all of the cycling mRNAs, you find the number you get from liver, then from kidney, then from spleen, heart, lung, et cetera, and you add all that up, you get at least 70% of the genome of the 22 or 23,000 genes we have, which undergo circadian oscillations somewhere in at least one tissue. So it's very simple. If 70% of our genes go like this in a circadian manner somewhere, is it any wonder that almost all of physiology is impacted by the clock, right? So that's the, of course, the devil's in the details there. You can imagine all kinds of questions you would have, but that gives you a sort of broad picture. So let me say a word about metabolic diseases, for example, diabetes. And there's evidence. I wouldn't call it conclusive, but intriguing evidence that the clock, circadian rhythms, have a big impact on metabolic disorders. Probably the strongest evidence comes from Shiftwork, which has a very big impact on a very high incidence of metabolic syndrome or diabetes. And the way to think about this, I think, or at least an important feature to keep in mind, something that's a little unexpected, wasn't unexpected to us in the circadian community when it was discovered less than 20 years ago, is that food metabolites entrain peripheral oscillators. And when I say peripheral oscillators, I'm referring to liver, kidney, all the stuff here, as opposed to here, right? So the general picture for mammals, the way everyone thinks about this, and I think is largely accurate, is that there's only one way for us to get light information, that is to adjust our clocks every day to the light-dark cycle, and that's through the eye. And if someone is interested in the question period, ask me about blind people, because it makes for an interesting story. But suffice it to say that this is a part of the brain, and the only part of the brain which extends out outside of this football helmet that evolution has provided us with, and not quite good enough for American football, I must say, and so that's the route into the brain, and there is a direct connection to the brain, to the suprachiasmatic nucleus, from the retina hypothalamic tract. The optic tract goes right to the right place in the brain, and all of that then connects to the retina hypothalamic, HRT, the adrenal gland, glucocorticoid cycle, and so there's a whole top-down view of how all the peripheral oscillators are kept in sync. And then two labs in the early 2000s did an experiment, a very simple experiment. The wonderful thing about biology, sometimes very simple experiments are incredibly insightful. And they simply fed rodents exclusively on the wrong time. That is, they were only given food during their sleeping time, and all their peripheral clocks shifted in sync with the availability of the food, despite the fact that the light cycle hadn't changed, and a totally top-down view would not have predicted such a result, right? So normally everything's in sync because our brain is also affecting behavior and appetite and all that kind of stuff, and so we eat at a time in sync with the brain. But if you deliberately shift the food, then all of the internal clocks are shifted, which means that you can see in light of that experiment why metabolic syndrome might arise from circadian or sleep disruption, people eating at the wrong time of day for one reason or another. And so this sort of introduction leads to what I think is a very pedagogically useful paper, if not medically useful. And there have been follow-up studies on this which have repeated the general idea, and it's very simple. If you take two rodents and you one of them is fed ad lib, that means they eat whenever they want, as much as they want, which is the standard way that laboratory rodent experiments are done. And the second population, you restrict the feeding to their active phase. Now, this is not the weird wrong time when they're sleeping. This is when they're up but rodents don't eat exclusively only at that time. And now if you make them eat only exclusively at that time and feed them high-fat diet, you can see from the photo, the ad lib fed animal gets obese, and the time-restricted fed animal does not, and they consume exactly the same number of calories. Exactly the same number of calories. So the principle is that what's important is not just how much you eat and what you eat, but also when you eat. And to put this in human terms, or sort of two examples, it's probably not good for you to go to the fridge at 10:30 or 11 at night and have a snack before bed, because your body is shifting its metabolism. All your peripheral organs are sort of going through a mini fasting cycle coincident with sleep. And the sort of second human part of this is I've been doing this myself now for about 18 months. That is, not eating for eating for 10 hours and not eating for 16 hours. And I've lost weight. Of course, I probably consume less calories. Lord knows if it really makes any difference. But the point is it makes this experiment is very compelling with the rodents, and it makes biological sense. It keeps in sync with the shifts that are naturally happening in the periphery. Okay, so let me segue a little bit to sleep and talk about this particular problem and why I think it's interesting. And of course, sleep has been of interest to circadian biologists for a long time, because we've known that the clock has a major impact on when we sleep, when we're alert and when we're asleep. So the clock is driving cycles of sleepiness and alertness, and that's one major input into sleep and activity rhythms, human sleep and activity rhythms as well, along with homeostatic regulation. So homeostatic regulation is super interesting, and it is the concept that there is a counter or a timer which is keeping track of sleep debt or sleep need. In other words, how much sleep you've had and how much sleep you need. And we really don't know what that is, and it is at least in part a separate system from the circadian system. So this brings me to this slide and this challenge, if you will, understanding the basic science of sleep. Who is keeping track of sleep need? How does this sleep homeostatic mechanism actually work? And by who, I don't mean, your mother, or, you know what I mean, right? So, and then more generally, why do we sleep? What's the function of sleep? And Professor Nedergaard here in Copenhagen has had a major impact on how we think about this problem. But I would suggest that this is not a done deal yet, and we really don't have a single agreed upon function of sleep. And by that I mean, by analogy to the circadian story, something which is true for flies and for mammals, a mechanism which is shared between the two species. And I'll say a bit more about that in a couple of minutes. So let me, before I say more words about flies, let me say something a little more about us in this context, and about larks and owls, early birds, morning birds, early birds, and night owls. So our rhythm, when you measure it in humans, is 24.25 hours. We run about 15 minutes slow every day. And you should ask me in the question period how that is actually measured, because unlike the rodent, which can be measured in constant darkness, that's an unethical experiment. You can't put a human for 12 days or something in constant darkness. So there's a very interesting runaround that's been devised in order to get this number, to figure out what this is, and I'll explain it to you if you're interested. So here's a consequence of or I should say a picture of something that Till Ronenberg, a circadian colleague in Munich, has named social jet lag. And what you see there are, what, 1, 2, 3, 4, 5, 6 weeks of data in which an individual has kept track of their sleep log profile. And this is an owl. This is somebody who likes to go to bed late and wake up late. And of course, because these people, owls, just like everybody else, have to get up for work or school at 6 or 7 in the morning, they are more impaired than early birds or than the average person because they're more sleep deprived and their circadian rhythm is more out of whack with respect to the rest of the system. And you can measure that and see it visually very clearly by looking at the 2 green days after the 5 hatched reddish days. And of course, that's the weekend. And so what you notice is that there's 5 profiles, and then of course on Saturday and Sunday morning, or Friday night through Sunday morning, those two days, that individual likes to go to sleep really late and likes to sleep late because they have a bigger sleep debt than average, and certainly than an early bird. And in fact, when you look at how much social jet lag individuals have, that's what's the dislocation between the weekend when they're sleeping by preference. The weekend sleep is how they would like to do it, and what they have to do during the week. Then early birds suffer less for this. So that's just a little bit about humans in order to perhaps stimulate some questions. And so let's return to flies and this question of why we sleep and what's the conserved function of sleep. And Torben stole my thunder a little bit because I want to extol the virtues of flies here in a general sense. And although the Nobel Prize is limited to 3 people, nobody ever said that an animal couldn't be a co-recipient, even if that put the number at more than 3. This cartoon was drawn by Mike Young, who's the blonde guy on your right, from Rockefeller. That's his great nephew who drew that cartoon. So all 3 of us are great fly chauvinists, and I need to point out to you that this was the 5th Nobel Prize. Is that more than Denmark has? And anyway, so I think, by the way, this counting of Nobel Prizes is silly, right? But this is the 5th Nobel Prize for fruit flies, starting with the great T.H. Morgan in 1933, and so the fly has really played a prominent role in science in the last 110 years or so. And so now going back to sleep, I'm going to simply assert, without going through any of these in any detail, maybe I'll speak about one for a moment, that fly sleep is almost indistinguishable from human sleep. The only criterion which is hard to apply to a fly is an EEG, right? Because it's really hard to put stuff on the head of a fruit fly and measure brain waves. Although there's been a surrogate that's measured fairly recently, and it looks like something very similar is going on inside the fruit fly brain. And I think what's maybe of interest to some of you, those of you who are approaching my age, is that aging has almost exactly the same effect on fruit flies sleep as it does on human sleep. For example, early morning wakening, multiple wakenings at the equivalent of 3, 4, 5 a.m., for example. Those of you who are older may have experienced something like that, or you've had your grandfather tell you about it. So everything is almost exactly the same. And there are human families in which single Mendelian lesions have been found to be responsible for sleep syndromes. And those turn out to be the human equivalent of 4 of these original fruit fly genes. And they're responsible for short sleepers. They're responsible for delayed sleep phase syndrome, people who can't go to sleep early. They're responsible for 2 families with advanced sleep phase syndrome, people who can't stay awake past 7 or 8 at night and then sleep a perfectly normal 8 hours, but of course their phase is not in kilter with work, school, all that kind of stuff. So this is, if you will, a real blending of the fruit fly genetics and human sleep syndromes. And just to broaden the landscape a little bit, flies are being used to model and study a number of diseases, and the ones in bold are those that focus on the nervous system and the brain, in which a lot of interesting work is being done and a lot is being learned. And I should say that fruit flies, from the point of view of normal behavior, not disease, have a very, very sophisticated behavioral repertoire. In other words, they're not just your grandfather's Oldsmobile, right? Learning, memory, aggression, motivation. You can read these yourself. All these phenomena, which are of interest to us, addiction, it is very easy to see these in fruit fly population and use them to study the basic science of these processes. And what's amazing about this is that Drosophila does all this sophisticated behavior with a brain that has about 100,000 neurons, or 1,000,000 times less than humans do. So in other words, this tiny fruit fly brain is able to orchestrate very sophisticated behavior, and I may at the very end, if I survive, or we survive, touch on an explanation for this. And so I'm bullish on, still bullish on fruit flies, because the efforts at Genelia Research Campus are about to complete a wiring diagram of all 100,000 neurons, all million or so synaptic connections between all of those neurons. And of course, this is a much, much more complex nervous system and behavioral repertoire, for that matter, than its genetic competitor, if I could call it that, C. elegans, or the worm. And so, given the tools that exist today, the toolkit that exists in flies, I'm confident that a lot of progress will be made. And so in the last 5 minutes or so, I'm bang on time. In the last 5 minutes or so, I want to just say a few words about what one project that my lab is interested in, and try to convince you that this is an interesting problem. So in the fruit fly brain, in that brain of 100,000 neurons, we only see clock gene expression in about 75 pairs of neurons. What's shown here is one side of the brain, and in red is the expression of the period protein, one of these famous proteins. And other than glia, support cells in the brain, there's only about 1 cell in 1,000, which is positive. And so this raises a question for us. How come? In other words, I told you in the introduction, or as I went through the talk, that there's clocks everywhere, in every tissue, in every place in the brain, everywhere. And it is possible that the brain is an exception. That is, there certainly are clocks in the brain, but it's not clear that every cell, or even most cells, in the brain actually have a clock. And so this is something we're interested in exploring. What's the definition of a clock neuron, and what fraction of fly brain and even mammalian brain neurons actually have a clock? And so these 75 pairs of neurons are responsible for generating the behavioral pattern that Konopka and Benzer originally assayed. This is the pattern of fruit flies in a light-dark cycle. And what those fruit flies do is they're active in the morning, they slow down in the middle of the day, they're active in the evening, and then they sleep at night. And for those of you who, I don't know, does Denmark have mosquitoes? You guys are too clean living to have mosquitoes. In the East Coast of the United States, mosquitoes, if you have a cocktail at 7 at night, you have a big mosquito problem. But if you're fortunate enough to have a cocktail at 2 in the afternoon or 2 in the morning, you have no mosquito problem. And this is the reason for that. That is the pattern of insect behavior. And those 75 neurons are responsible for orchestrating that behavior. And so this small circuit, 75 pairs of neurons, is responsible for governing this sleep and activity or sleep-wake pattern. And of course, it gets internal input and it gets environmental input, light, for example, as a way of dealing with this. And we like to think of the mammalian suprachiasmatic nucleus, the region of the hypothalamus, which is the most important center in the mammalian brain, the equivalent of these 75 pairs of neurons as a poor man's fly clock system. Now, the mammalian people take umbrage at this view, of course, but I think it's fair to say that a simpler system is more likely to yield interesting results. And as I've said before, and this is probably a little out of order, the eye directly connects to this region of the mammalian brain, the suprachiasmatic nucleus. So I'm going to tell you in 2 or 3 minutes, or 4 minutes, some contemporary experiments which address the question of how heterogeneous are these 75 pairs of neurons, and this is done by single-cell sequencing. And importantly, for those of you who know a little bit about this, this is done as a function of time. In other words, the sequencing is done by taking brains that are at different times in the circadian cycle, and almost all single-cell seq results in the literature are single snapshots. And in so doing, they are potentially missing rich heterogeneity of gene expression, which comes from looking at something more dynamic, the presence of a male in the cage, or something which changes the way the brain might respond. And so these experiments have been really spearheaded by one postdoc, Dylan Ma, and I want to just come to one sort of experiment, which is sort of the key result, which I think will frame a couple of the more general points I made. So the way this experiment is done is to have a tool in the fly brain which expresses in most of those 75 pairs of neurons and very few ectopic cells or very few other cells. And therefore, with those neurons labeled this way, we can actually capture those cells then and sequence them and ask the question, how heterogeneous are those 75 pairs, of 60 in this case, because we capture about 60, how heterogeneous are those neurons? And the answer is that there are 32 well delineated individual clusters as defined by gene expression among those 60 pairs of neurons. And they all have marker gene expression which delineates them carefully. And what is remarkable here is how heterogeneous this small set of neurons really is. And at least 16 of those clusters can be unambiguously assigned to particular regions of the fly brain based on the immunohistochemistry results that exist in the field for the past 20 years. We know what markers this particular set of cells makes and this particular set of cells, and that allows us to assign particular clusters to particular cells, which is then assigned to particular functions. So I'm going to summarize and simply say there's this remarkable level of heterogeneity, and I think it's much more heterogeneous than the sequencing results from a mammalian brain would indicate. And I think that offers a clue of how a brain with 100,000 neurons can do these sophisticated tasks, because those 100,000 neurons are operating in groups of 1, 2, or 3, the equivalent of something in mammals that's done by a much larger number of neurons for reasons that we don't completely understand. But I think the sophistication comes from the fact that we're dealing with an extremely heterogeneous brain. And our interests, as it says, are twofold: gene discovery and function, and the neuron-specific regulation of gene expression. And I think a goal to end on a note of sleep is to really being able to assay the entire fly brain under specific conditions of sleep deprivation, under particular conditions, and to try to see which cells are changing in exactly which way, to try to perhaps be able to tease out what is the real function of sleep. And with that, I'm going to thank you once again for the invitation, for the attention you have all paid, and for thank Providence for having let me survive this despite the jet lag. Thanks very much.